In this video, I will be showing the effects selection has on a population of snails. These snails are all similar except for the appearance of spots. This is due to one genotype that is associated with the phenotypic spots. These snails can either have bright spots, light spots, or no spots. Since the snails have been living in an environment with little pressures, the population has arrived at the hardy-weinberg equilibrium. This means the population is not evolving and is not affected by genetic variation in natural selection. To begin, I collected 100 snails and noted whether they had bright, light, or no spots. Since we already know how the genotypes are related to the phenotypes, we can determine the genotype frequency. You just divide the number of individuals by the total. To find the allele frequency, you add half of the heterozygous genotype frequency to each of the homozygous genotype frequencies. We can use this method because for each genotype we have two alleles. For the 32 bright-spotted snails, we have 64 big B's, and for the 54 light-spotted snails, we have 54 big B's and 54 little b's. And for the no-spotted snails, we have 28 little b's. Another way to check your calculations is to use the hardy-weinberg equation. Just plug in the allele frequencies previously found. Now, sadly for the snails, a new bird species has been introduced to the environment. Now the snails feel the pressures of natural selection. The spots on the snails make them an easier target to see from the air. Very quickly, we are able to see that the snails with no spots have a better chance of avoiding detection from the birds. This kind of selection is an example of a directional shift. This is when a population evolves towards one end of a trait spectrum, in our case, no spots. The birds cause a drastic relative fitness effect on our spotted snails because most of the snails are eaten before they can reproduce. This decrease in spotted snails affects our future phenotypic ratios, so now we use the genotype frequencies and relative fitness to calculate the new phenotypic ratio. First, we calculate the survivors by multiplying the genotype frequency by the relative fitness. Since survivorship is affected by the relative fitness, our total no longer equals 100%. To get a more reliable measurement, we divide the survivors by the new total. After this, your population now equals the correct 100%. From this, you can see that the frequency for bright-spotted snails decreased from 32% to 14%. The frequency for light-spotted snails decreased from 54% to 48%, but the frequency for no-spotted snails increased from 14% to 37%. This change in genotype frequency is due to the no-spotted snails being able to escape most predation from the birds and living to a reproductive age. Since the snails with spots are more easily detected, a majority of them will not reach a reproductive age and be able to pass on their genes to future generations. When you take the new genotype frequency and calculate the allele frequency, you see the numerical difference between the before and after selection. The allele frequency of our big B decreased, while the frequency of the little b increased. You also see this effect when looking at the graphs that show the directional shift. If the birds remain in the environment, eventually the bright-spotted snails will go extinct, while the light-spotted snails will struggle to remain alive. In this scenario, the no-spotted snails are the victors.